Hello, this is Thomas with DNASTAR, and today we're going to be going over the new protein impact column and variant analysis workflows that we just released in the MegAlign Pro 17.4.2 release. Now to get started, I've launched the DNASTAR Navigator. I'm going to click on the Organize by Application button, which brings up all the applications we have in the suite in a list. Then I'll go ahead and launch the MegAlign Pro tool. Now to get started, I'm going to click on New Project and New Blank Alignment Project in MegAlign Pro. Next, and I'm going to bring in a GB file of the HU1 strain of COVID and also 334 other additional randomly selected strains that I got from NCBI. Now, if you notice, these files are in GB and GBK format, which is really important for the workflow I'm showing today, because that includes gene and CDS features. And with those gene and CDS features, we can look at nucleotide data and give you information on how the codons change and what proteins are also being changed from the codon changes. Now, I'm going to click on this strain here, the HU1 strain, and I actually want to rename it with fields. So I did a right click, I'm going to go to rename with fields. And then I'm going to take the default name out, and then I'm going to click on description and then add the description as the name. And this will be the name that's now given to that reference. Now to set it as a reference, I'll right click on it and use as reference. Now we're going to benefit this alignment by using the MAFT aligner, and the MAFT aligner has a new reference-based multiple sequence alignment capability. Without using the reference-based alignment in MAFT, this would take hours using the other aligners. But because of the MAFT alignment, this tool now will be able to finish this multiple sequence alignment in less than a minute. Now, one of the first things I always do after an alignment is I go ahead and I build a tree out of the alignment. So we have this tree button here, which I'll click on. We have two options for trees. We have neighbor joining trees and maximum likelihood trees using RaxML. Now RaxML maximum likelihood trees also provides us a way to do bootstrapping and have confidence values on our clades, but it takes much longer. For this demo, I'm going to use a neighbor joining tree instead. Now while that tree is being calculated, I'm going to open up this window a little bit, and then I'm going to click on the compute variance button. And what this tool now will do is go through the reference and compare it to all of those other sequences that we've inputted, and then give us all the nucleotide changes. But not only that, it's going to give us codon changes, synonymous, non-synonymous calls, including frame shifts, and it'll tell us which protein has been mutated and which residues it has been mutated to from the codon changes. Now, one thing I'm going to click on here is a detach view button, which detaches the view and then lets me maximize it. Now, if you notice, we have all the variant positions and the referent positions listed here, and all the different sequence identifiers on the left side. We also have an impact column and a feature name column, and this impact column lets us identify things that are non-coding, that are synonymous calls, and non-synonymous calls. Now, one interesting thing we can look at now is we can go down to the spike protein, and we can quickly look at all the differences that were found in this data set that I have. So here's the start of the spike protein features. You can see there's a number of non-synonymous calls. At residue 19, there was a call. And if we scroll down further, at residue 142, there was a call. At residue 156, we have a call. At residue 452, we have a call. Residue 478, residue 614, and also residue 681. Now, additionally, including the residue and codon changes that are occurring, we also get changes in charge, changes in PI, changes in hydropathy, changes in aliphatic index, and changes in instability index. So these changes are actually looking at the gene and CDS features of the nucleotide changes. Because it's non-synonymous, we have a protein change that occurred, and we call this the protein impact columns. And this can give us some information on how the protein has been perturbed by these changes that we have found. Now I'm going to click on the reattach view button to get this reattach into the view, and it looks like our tree is almost finished computing. So now that we have a tree, we can scroll down through the tree, and I want to see if we can find our reference that we use, because that's the baseline for this tree. And there it is, and it was handy for me to rename it because then I can easily identify it. Also, we can change the color of the tree by clicking on this button right here, and then I'm going to change that initial clade to red, and that's where the start of my comparison was. Now, one thing that we can do now that we have this tree is we can do a control click on individual parts or clades of the tree, and then a right click, and then we can quickly get a pairwise alignment based on those two sequences. And we can do a local Smith-Waterman or a global Needleman-Wunsch pairwise alignment. Now in this case, I'm going to do a global Needleman-Wunsch because I want to make the best global alignment, which really tries to fix the endpoints. Now, if you notice, we also get information on this pairwise alignment. We get identity, we get the percent gaps, the number of identical nucleotides. And then we can scroll down and start looking at the differences. And here are some of them. Now, one handy thing you can do is if you do a click and then a shift click, and then you do a right click and a copy, we can easily get this into a format that can be pasted into Word or Notepad, and has a really simple format. Also, it's got a gap percentage, and it's got the number of identical residues. It also has the identity of the alignment. Now, one of the things the tree also provides is the tree provides us an order to the sequences. But if we go back to our sequence list, the sequences were put in a random order. And so one of the things the tree lets us do is if I go to the tree menu, I can order the sequences like the tree. And then that flips all the sequences around so that they're in the same order as the tree. And this can help us identify and select and view sequences that are more similar based in the sequence view. Also, we provide a distance table of all the sequences so we can see a distance table comparison that's color coded, and we can change some of the values here, but we have access to distance, percent identity, percent gaps, the identical value, the gap count, the gap length, and the length of the sequence, all in this distance table. So these are some of the latest features in the MegAlign Pro 17.4.2 release, and if you guys have any additional questions, feel free to reach out to me, and we can provide additional training or help your team use this workflow with the data sets that you're working on. Thanks a lot, and have a great day.